my dear friends, and thank you very much for invitation. I'm from Riga, as it was said, and I have a rather long experience in science. And what I want to say, there are two problems that are engaging me through all these years. This is cancer and organization of cell nucleus. And in the very interesting way, the both problems, one problem is cancer that is very difficult to treat, and it is a resistant disease to treatment, where still the cells are surviving at the brink, and it is our last review just published on that. And also the issues of the genome regulation, where I was always interested in the whole genome regulation, in this in biophysical aspect, I should say. So there are somehow converging problems. There are fusing, and there are fusing on the base of thermodynamics. So what does it mean for cancer research? For cancer research, now we're coming to recognition of the priority of epigenetic over gene-centered genetics. What I mean, not epigenetic in the narrow sense, but in the wide sense that was introduced by Conrad, was introduced as the hill, epigenetic hill, Waddington Hill by Conrad Waddington in 1957, where he introduced such a metaphor that a zygotic cell is on the summit of this hill with the highest energy, and to become differentiated, the cells should go along these values, and differentiated cells have lower energy and are at the bottoms of these values. And so in general, it means that cancer can be reprogrammed close to embryo, and current data show that full-blown cancer metastatic has the transcription expression profile close to two-cell embryo, and what is most important, that it is multipotent, and this multipotency is can do over genetics. And at the same time, it's also recognition that the cancer cell can be in principle differentiated by epigenetic means. And in fact, both notions were introduced by Stuart Kaufman 50 years ago, and here I just praise him. So what is needed for such changes? Cell fate change is needed, and it must be reached by cannot be reached by linear thermodynamics. It needs a jump, and it needs critical self-organization of transcription pattern, so bistable switch. And for chromatin research, it means that we are speaking more and more about self-organization, physical aspects. It needs critical self-organization and also the instruments to govern it. And I repeat that we are interested in the whole genome studies. So the instruments may be also heterochromatin, and that is what we are interested in. So the model is very well known to you all, maybe many of you MCF cells. This is breast cancer, it is luminal type. It is not quite undifferentiated, it is a bit differentiated. So in the Waddington Hill, it is located somewhere in the middle. And the model that we are using is differentiation by growth factor. It is neuregulin or huregulin called, that can make these cells to produce milk it takes time, 14 days. What we are interested in, the very first changes, how the cells come to this decision, how they take this decision when it becomes reversible. And previous studies showed that it is attained in the first hour. And so we concentrated our studies, and we're interested in heterochromatin on the first 60 minutes, a little bit more. And so we were basing also on the transcriptome studies of this model that was reported very much in detail and dynamic by Japan groups from Hatekayama laboratory, Saeki and Chelios. Then it was also studied by a Chinese group published in 15, and quite along by the group of Masachuchi and Kenichi Yoshikawa, several articles where I also a little bit participated as a biologist. And what we decided, we need to repeat this model, its main hallmarks, to be sure that we are working with the same model. So we treated control cells. They are a serum start for 48 hours. We treated them with heregulin 50 nanomoles. And after 60 hours, we observed the expected clustering of RB2 receptors. In fact, what is going on, RB2 receptors are dimerizing with RB3 receptors cluster, go into cell nucleus, and finally start in 5 minutes already 2 signaling cascades that are needed for this differentiation commitment. So we concentrated on this first 60 minutes. So what was earlier showed on this model of differentiation commitment? As I mentioned, the group of Masachuchi, and it was just we were motivated by these studies. So what he found, he took this transcriptome data, and these are 22,277 genes in dynamics. And he studies the degree of temporal variation in expression for each genes and grouped them in the genes of high variancy, intermediate, and low variancy genes. And what he found, that there occurred an avalanche of all genome transcription involving also this static group of low variancy genes. And it occurred precisely in 5 minutes, between 15 and 20 minutes after heregulin action. And so it was occurring like an avalanche. Yeah, usually it is called avalanche, like a lavine in the mountain. All genome has changed and accelerated its transcription. It was seduced later after some time. So the same was studied by Saeki differently. Saeki paid attention to BI-PHASIC early stress response that is occurring at the same time. Just observed several genes of FOS group. That's very important, stress response genes. And they found such a dynamic which we try to reproduce. So in their dynamics, FOS transcription factor, it was activated practically from 0 to maximum 20 minutes. Then it is activating FOS ligand 1 activated at maximum 60 minutes, and so on. And this BI-PHASIC stress response, very high, transcription is afterwards undergoing downregulation feedback by the factors that are also acting in these groups. These are two genes, FHL2 and DIPA. So we did qPCR in our obstacles to show the same, that indeed FOS is activated in 20 minutes, FOS ligand 1 in 60 minutes. We also studied CMYC, that's important, and also at 60 minutes with maximum. And afterwards, at 3 hours, as is shown here, they went down. So we reproduced these main hallmarks, and so we could study what we wanted to study, heterochromatin. But here I want to mention one thing that was highlighted both by Saeki and also Chuchia. In this model, they also compared the action of heregulin with ergosterol, the other transcription factor. And this other transcription factor does not cause commitment to differentiation. The cells continue just to proliferate. And the difference was that heregulin caused sustainable high expression for at least an hour and a half. And the same notion, in fact, was deduced by deep studies of Masa Chuchia from this transcriptome dynamics, that there appears some energy engine that is needed for the cells to go to this point of decision. So now we suggested that constitutive heterochromatin, pericentric heterochromatin may be somehow involved in this leap of transcription, and just studied it at these time points. So we stained the cells with label for repressive histone mark for heterochromatin, also for centromere mark to show that they really, these pads make these clusters that include centromeres. You see these clusters of different size, larger are near nucleoli. And for our measurements, we had to know where in the cell cycle we are, because we want to study the sizes and amount of these particles. And we are in the starving cells, mostly by 85% in G0, G1 cells are, some still continue proliferation and entered mitosis low percentage, and this could not be stopped in these cells. But still we could reckon with this 85%. And we also showed in these experiments at once, these are single cells labeled by dots, that they have very variable average number of these pads, chromocenters per cell. So next we studied the relationship between the number of centromeres and the size of pods, their area, and found fair linear correlation, that just confirming that the centromere cluster pods, larger, smaller clusters. Then we found the most interesting thing. In the ImagePro software, on this confocal pictures of maximum projections, we studied both in averaged cells or individual cells, we studied the relationship between the number of pods and their area. And we found a very nice negative exponential correlation, or even power law, both for averaged data, and these are for individual pods, where 570 was measured in 22 cells. So it was very interesting revelations that the relationship between number and size of pods obeys exponential decay or power law, which suggests that they're acting as a dynamic integrated system, which can split and fuse the constant amount of this same constitutive heterochromatin. And also from statistical mechanics, this relationship, the absence of any Gaussian distribution, just this scale-free distribution, shows that they are capable to synchronize by bursting. And this can occur by all or none law in a jump. So this is unstable thermodynamics. So what Herigulin is doing, we're very interested to look for these 15 minutes or between 15 and 20 minutes when Chuchie found this huge avalanche of the whole genome transcription. We found that pods, the black is a heterogeneous distribution of the control, it's a base power law distribution, and pods were shrinked to one spot, lower than 2 microns, and increased in number. So they were splitting. And here in box plot, you see the control, larger average amount and rather heterogeneous distribution of these chromocenters, while after Herigulin, they come into one comfort attractor of smaller pods with very low variance. Observations also show that this bursting of pods was rather directed from the nucleoli, where around nucleoli larger pods remained less changed, and we see such chains of unraveling towards in the radial position. So what about transcription of the heterochromatin itself? So for that, we just were curious to see for satellite 3 primers, there are this transcription is specific for each chromosome. So we got primers for chromosome 1, chromosome 9, and looked in PCR analysis. So control is green, and after 15 minutes, we have decrease statistically reliant, but not large, 20% for both probes. And at 60 minutes, we found 1.5-fold increase of this transcription activities. And in fact, the epigenetics of pods, which we started to study a little bit, it is also acetylation is decreasing here, but it needs more studies. So what about euchromatin? Euchromatin was stained with the specific histone probe, trimethylated in a K4 position. And what we applied in image pro-analysis, we applied such a program of heterogeneity that shows the change of the distribution of the label of the whole nucleus if it is different by 10% from the average. And we found this is control that the heterogeneity halved. That means that the euchromatin was becoming more homogeneous, more extended. We did some pilot experiments, one-line densitometry through the spots in control, in uregulin treated cells. And the cells were stained by 3 labels, it is DAPI, and for red for repressive heterochromatin, and green for active chromatin. And what we found, to say in general, that euchromatin is repulsing from pots after 15 minutes of uregulin treatment. That is correlating with decrease of heterogeneity or increase of homogeneity. We also applied such an interesting analysis, DNA confirmation test, acridine orange, with which we were working with for many years. And this acridine orange is metachromatic fluorescent dye, very specifically binding nucleic acids. And if we apply RNAs and we apply it, so it is specifically binding DNA. And it is binding DNA in 2 mechanisms. One is intercalation between nucleotide bases, and it gives emission green at 530 nanometers. And the other is the ability of this dye to bind to site-free phosphate groups as dimers, polymerizing and giving this metachromatic shift with the emission at maximum at 640 nanometers. So very simply, we used the epifluorescent microscope, we used filter, I3 filter, and we imaged the cells control after 10 minutes uregulin action, 20 minutes, 60 minutes, very quickly, many cells, because we wanted to see the behavior of the population. So individual cells were measured, and the obtained values were converted as in gray scale and measured as integral optical density. And we separated these channels, these images in green and red already in computer. And in this way, we calculated ratio red to green. Yeah, that is characterizing unfolding or conformation of the DNA. And what we see, starving cells are this green, yeah, and 10 minutes, no shift. And at 20 minutes, we have shift, increasing the relationship of red to green. That means that DNA has undergone some degree of unfolding and can bind these polymers of acridine orange. It was very constant in all experiments, but the next shift that was occurring in 60 minutes, further unfolding, it was variable, it was depending on the population experiments. So in this way, case, as you see, we have 2 peaks. One is remaining with the first shift, and the other, some part of population, about half, is undergoing more DNA unfolding. So it is another experiment, all was shifting, and in some experiment, we did not get the second shift. So this is probably this engine, energy engine that is needed for the cells acting as a whole population or characterizing the energy level of each of them maybe to go to this state. So anyway, euchromatin was unfolding in 20 minutes certainly, and in 60 minutes in many cases. This is in fact what we came to the hypothesis that acceleration of transcription, critical acceleration of transcription of the whole genome that was caused by heregolin in a jump critically in between 15 and 20 minutes may be caused by splitting of pads that also show this critical shift to smaller particles. And maybe in this way, these pads constituting heterochromatin is coming under critical threshold of silencing. So silencing is taken off. What is going with the cells if we are speaking in the terms of Waddington-Hill? The cell is starting the way to differentiation, commitment to differentiation, but it is jumping up in this hill. So it is going in the state that is more close to the stem cells. And it is known from literature that embryonal stem cells have small pods. We are now speaking a little bit about this BI-PHASIC gene response of stress response that I mentioned to you. So the 20 minutes, we have erasement of the pattern by paths. At 60 minutes, we have maximum phospholigand 1 and also maximum CMYC that would cause open chromatin, most open chromatin, acetylation of histones. And from literature, we know that phospholigand is rather an universal adapter of differentiation, acting in many kinds of differentiation. And it can hook here estrogen receptors that is available in these cells, and so start this differentiation, repatterning that will occur later. So instead of conclusion, we are speaking about cell fate change in cancer cell, differentiation commitment, just in these cells, MCF cells. And we see many signs of nonlinear thermodynamics of cell fate change. Between them, biphasic early stress response. These are bivalent genes, so they have bivalent chromatin, it is known, can very quickly change. Most of them, phos and phospholigand, have disordered protein domain at the end. All this is nonlinear thermodynamics. Then we see scale-free pad bursting, which is erasing silencing pattern occurring also by critical in a jump. Then we have transcriptional critical self-organization described by Chuchia. This is genome avalanche. It is also nonlinear thermodynamics, and it is creating energy engine. And I forgot to say that in the article of 15, the genes were specified as groups that are reacting in this avalanche with a medium size 1 megabase DNA domains found in old chromosomes. So these are TATs. So what we have bistable switch, and we have this differentiation commitment of these not fully differentiated cells. MCF cells, they must go to the higher energy level to the more multipotent state, to be able to go into the next well of repatterning of transcription differentiation in later days. So we have published it in a biophysical journal in January of this year, and we are very thankful to financial support of our Nucleome Consortium, who paid for this publication. And here is our team from Riga, from our institute, from university, and two master students, in particular Jakob Kriegers has done a lot of this part of this work, Felix Rumnieks, Kristina Salmiņa, molecular biologist Inna Inashkina, bioinformatician Pavel Zayakin, and my long-standing collaborator, optical physicist Freivalds. And here I want to acknowledge our vice physicist, biophysicist, and system biologist Giuliani, Masa Chuchia, Professor Kenichi Yoshikawa, and long-standing collaborator on the chromatin, Professor Michael Hausmann. And thank you for your attention. And I'm open to your questions.
I was intrigued by this, large-scale changes with the pericentromeric regions clustering around nucleoli, and I was wondering if that could be due to differences in activity in nucleoli themselves, right? If the cells have to all of a sudden have an avalanche of protein production, you can imagine that they have to make many more ribosomes, and that the nucleoli transcriptional activity will increase. And so are these changes in the pericentromeres actually caused by increased activity in nucleoli?
Yeah, no, we have not studied it directly, but in fact, what we are now, we are writing a large review on transcription pulsing, in fact, because, this scale-free particles, they should act as a network and as a pulsing network. And in this review, I give some other observations showing that this pulsing is going from nucleoli. You are right. Yeah, and even the very old observations of Boveri, who called the nucleolus a heart of the cell, because he found this pulsing extrusion of vacuoles in very active, in transcription active cells. So nucleoli really is central to this system. Mm-hmm. Thank you.
I was curious about this these pads. So HP1, I guess, is involved in the formation of these pads. And did you see changes in the HP1 behavior during your activation, or or change in phosphorylation of HP1 or something like that that may change the biochemical properties of HP1 that may drive this change in the organization of the pads?
Yeah, that's a good question. We have not studied it, in fact, much. We have not studied it, and we had a very strange HP1 antibody which we tried. It stained nucleoli only.
Okay.
So we stopped with that. But of course, to study the epigenetics of pets in this situation is very crucial. And we have some plan. Yeah, we have some plan, because we also noted that they are extruding some substance. Yeah, we shall study it. Yeah.
And maybe a related question is, when you have the activation, did you see change in the H3K9 density? I mean, or H3K9 is remaining at the same density, and it's just the pads that are splitting, or it's also the total number of H3K9 that is also reduced?
We have not measured it.
Okay.